Picture yourself walking through a crisp autumn forest.
Oh, the best time of year for it.
Right. The air is cool, the leaves are crunching right under your boots, and right there, just sort of emerging from the moss, you spot this beautiful golden brown mushroom.
Just waiting to be picked.
Exactly. It looks absolutely perfect for dinner. I mean, you can almost smell it sautéing in butter already.
Which is a very tempting thought.
It is. But what if that single, unassuming mushroom could tell you a story about a 1986 nuclear disaster?
Yeah, that escalated quickly.
Right. And not just that. What if it also held secrets about the deep earth chemistry of heavy metals? And just to top it off, what if picking it could entangle you in a complex web of modern American land laws that could legally land you a $20,000 fine?
It's quite the resume for a little fungus.
It really is. You know, you'd think foraging is just this relaxing walk in the woods, like a simple way to connect with nature.
A lot of people do think that, yeah.
But it's actually this massive collision of botany, nuclear physics, and legal mazes.
It really is a minefield.
So today's deep dive is custom tailored for you, the intellectually curious learner. We are taking a stack of scientific studies from Europe, regulatory guides from federal land agencies, and ecological field notes to unpack the fascinating, totally hidden world of Cortinarius caperatus.
Also known as the wrinkled quart.
Yes, the wrinkled quart. It's this widespread, highly sought-after edible mushroom that, well, secretly acts as an environmental sponge.
A very efficient sponge at that.
Right. And by the end of our time together, you won't just look at mushrooms differently, you'll understand the invisible chemical and historical records buried right beneath your feet.
Yeah, our goal today is really to go beyond just identifying what this mushroom is. I mean, identification is great, but we need to examine the underlying mechanisms.
The how and why.
Exactly. Why does it behave the way it does? How does it interact with the soil at a microscopic level? And, what does its unique biology teach us about the interconnectedness of our entire ecosystem?
So where do we even start with something like this?
Well, to truly understand Cortinarius caperatus, we have to, let's zoom out first. We need to look at the broader family it belongs to.
Okay, the Cortinarius genus.
Right. Commonly known as the webcaps. And the sheer scale of this family it's honestly staggering.
How big are we talking?
Over 2,000 known species.
Wait, 2,000?
Yeah. It is the largest and most diverse group of mushrooms in the entire world.
That is a massive family tree. I was looking through the ecological field notes, and honestly, navigating a genus that large seems like an absolute nightmare for a taxonomist.
Oh, it is notoriously difficult. But they do share some very specific, observable family traits that link them all together.
Okay, so what's the first thing you'd look for?
First and foremost is their ecological role. They form mycorrhizal relationships.
Mycorrhizal, meaning they interact with roots. Okay.
Exactly. They live in this highly evolved, mutually beneficial symbiosis with trees and shrubs. So underneath the soil, the fungal tissue, which is called the mycelium, wraps around or even physically penetrates the tiny rootlets of living plants.
Like pines or spruces?
Yeah, pine spruces or members of the heath family. The fungus basically acts as a massive secondary root system for the tree. It breaks down minerals in the soil and delivers water and nutrients that the tree's own roots could just never reach on their own.
Oh, wow. So they're literally feeding the tree.
They are, and in exchange, the tree photosynthesizes sunlight into liquid sugars. Then it pumps those carbohydrates down into the roots to feed the fungus.
It's a perfect trade-off.
It is. But because of this absolute reliance on a living host plant, you will only ever find a Cortinarius growing on the ground.
Ah, okay. So they can't eat dead stuff.
Right. They lack the specific enzymes required to digest dead wood, so you will never, ever see them sprouting from a dead log or a tree stump.
Well, that structural limitation is a great first filter for a forager. Like if it's on wood, it's not a Cortinarius.
Exactly. You can rule it out immediately.
But the defining physical trait, the one that gives them the common name webcap, is something called the cortina.
Yes, the cortina. It's a key identifier.
I really love the visual description of this in the botanical sources. It describes it as a cobweb-like partial veil.
That's exactly what it looks like.
So when the mushroom is young and it's just pushing up through the soil, it looks like a delicate spider web stretching from the edge of the cap down to the stem.
Yeah, and that cortina serves a really vital evolutionary purpose.
Protection, right.
Exactly. The gills underneath the cap are where the mushroom produces its spores, its microscopic seeds, basically. And when the mushroom is young, those developing spores are highly vulnerable.
Vulnerable to what, like bugs?
bugs, slugs, or just drying out from the wind. So the cortina acts as a breathable protective membrane.
That makes sense.
And as the mushroom matures, the cap expands outward, kind of like an opening umbrella. And that cobweb layer stretches until it finally tears and collapses.
And when it collapses, it leaves behind a very specific marker on the stem.
Yeah. Right, what the field guides call a cortinal zone.
Yeah, usually it forms this faint, delicate ring, and that zone almost always takes on a rusty brown color, which actually brings us to the ultimate fingerprint of the entire genus.
The spore print.
Yes.
If you take a mature Cortinarius cap, take the stem off, and place the cap gills down on a piece of white paper overnight.
You get millions of microscopic spores dropping onto the paper.
Exactly. And in the case of the Cortinarius family, that resulting pile of spores will always be a distinct rusty brown or cinnamon color.
Every single time.
Every time. And those spores actually fall onto the remnants of the cortina on the stem as well. That's why that cobwebby ring turns rust-colored as the mushroom ages.
It's like a brilliant built-in identification mechanism.
It really is. It makes confirming the genus pretty straightforward once you know what to look for.
But within this massive family of 2,000 species, there is a distinctly dark side.
Oh, very dark.
Yeah. Before we get to the edible mushroom we're focusing on today, we have to talk about the deadly cousins. Because the field notes are incredibly clear on this point, foraging within the Cortinarius family is not for beginners.
Absolutely not. It is high stakes.
There are species that are genuinely lethal, right?
Yes. You're looking at species like the deadly webcap, which is scientifically named Cortinarius rubellus, and the fool's webcap, Cortinarius orinosus.
Just the names alone sound like a warning.
They should. These mushrooms produce a highly specific, naturally occurring compound called orinolin.
Oranan, and what does that do?
From a toxicological perspective, it's a fascinating and honestly terrifying molecule. It's a bipyridine compound.
Okay, what does that mean for the human body?
Once ingested, it specifically targets the epithelial cells of the kidneys.
Oh, wow.
Yeah, it disrupts the basic redox reactions, so the energy transfers within those cells, and that leads to cellular death and ultimately complete renal failure.
That is awful. But what makes orillanine truly insidious, at least according to the clinical reports, is its latency period.
Yes, the latency period is the real nightmare here.
It's deeply unsettling. If you consume a mushroom containing orillanine, the severe symptoms, the actual shutdown of your kidneys, might not manifest for anywhere from 2 to 14 days after ingestion.
Up to 2 full weeks.
Right. You might experience some mild flu-like nausea in the first 48 hours, but that usually passes.
So you think you're totally fine?
Exactly. You think, oh, it's just a 24-hour bug. And then 2 weeks later, your internal organs just begin to fail.
And the medical implications of that delay are profound. I mean, in clinical toxicology, diagnosing a poisoning relies heavily on the patient's recent history.
Right, they ask what you ate.
But human memory is notoriously fallible. If a patient presents with idiopathic kidney failure on the 14th of the month, the attending physician is going to ask what they've eaten recently.
And the patient is going to remember, like, the seafood they had yesterday.
Exactly, or the takeout from the weekend. They are highly unlikely to mention a woodland mushroom they foraged a fortnight prior, especially since they didn't get immediately sick from it.
Right. The psychological link between the meal and the consequence is just completely severed by that time gap.
Completely severed.
This actually makes me wonder how the medical community ever managed to connect the illness back to the mushroom in the first place. I mean, trying to pin a medical crisis on a specific ingredient from 2 weeks ago seems like an impossible epidemiological puzzle.
It really does.
It's akin to trying to blame food poisoning on a sandwich you ate 3 Tuesdays ago. You just never make the connection.
It required rigorous, long-term epidemiological detective work. The connection was actually solidified following a mass poisoning event in Poland in the 1950s.
A mass poisoning.
Yeah. Over 100 people fell ill with kidney failure over a span of several years, all in the same geographic region.
Oh my gosh.
So public health officials had to painstakingly cross-reference the diets, the habits, and the foraging practices of the victims.
That must have taken forever.
It did. But they eventually noticed that all the victims had consumed what they thought were edible mushrooms, but they had mistakenly included Cortinarius orinosus in their baskets.
So it was just an accidental mix-up by locals.
Exactly. And the delayed onset is exactly why it took modern science so long to isolate orillanine and understand how it works. It operates on a completely different biological timeline than, say, acute bacterial food poisoning or other fast-acting fungal toxins.
That really highlights the incredible stakes involved in wild foraging. You're interacting with potent biochemical compounds that science is, frankly, still working to fully understand in some cases.
You're stepping into a very complex chemical laboratory when you walk into the woods.
But out of this massive, sometimes lethal family tree, there is one specific cousin that is safe, edible, and practically famous among foragers.
Our main character.
Yes, the star of the show, Cortinarius caperatus, historically known as the gypsy mushroom, but now widely referred to as the wrinkled Cortinarius, or simply the wrinkled court.
Let's look at its physical characteristics and how it stands out in the forest, because you want to be sure you've got the right one.
Right. The ecological sources provide a very clear morphological profile.
They do. When the wrinkled court is in its button stage, meaning it's young and unexpanded, the cap is coated in this thin, superficial layer of silky, white, hair-like fibers.
The literature often compares this to a delicate layer of facial tissue or Kleenex stretched tightly over the center of the cap.
Yeah, I find that Kleenex layer visual incredibly helpful.
It really is. It's a texture you can immediately imagine touching.
And as the mushroom matures and the cap broadens out, it takes on a yellowish brown or tawny hue, and that silky layer breaks apart.
Right, the surface of the cap becomes dry.
Yes, and it begins to wrinkle radially, meaning the wrinkles radiate outward from the center to the edges, almost like the spokes of a bicycle wheel.
Which is where it gets its name.
Exactly. That wrinkling is the defining feature and the origin of its Latin-specific epithet, caperatus, which translates directly to wrinkled or furrowed.
That's super helpful. Are there any other identifying marks?
Well, additionally, unlike many of its webcap cousins that only leave a faint cobwebby zone on the stem.
Right, the cortina we talked about.
Yeah. The wrinkled court features a substantial, thick, white, membranous ring on its whitish stalk. It is a much more robust remnant of its partial veil.
Okay, so it's got a real solid ring, not just a faint smudge.
Exactly. It's very distinct.
So, if you are looking to find this mushroom, the geographical data is incredibly encouraging.
It is. It's everywhere.
According to the Global Fungal Red List Initiative, which assesses species for their conservation status and risk of extinction, the wrinkled court is categorized as least concern.
Which is great news for foragers.
It's not rare, it's not endangered. In fact, it has a massive geographic footprint. It is widespread across the entire northern hemisphere.
We are looking at a distribution that covers boreal, subalpine, and Arctic zones from North America straight across to Eurasia.
So you find it in Canada or Germany or Russia.
Yep. You can find it thriving in coniferous forests among spruce and pine, or in deciduous forests hiding under beech trees, growing right out of the dense moss and decaying needle litter.
That's amazing. And, the scientific history of this mushroom also provides a fascinating window into how the discipline of mycology has evolved.
Oh, the naming history is a wild ride.
Yeah. For many decades, scientists did not classify it within the Cortinarius genus at all.
No, they didn't. They had it in its own distinct genus. It was known as Rosites caperata.
And why was that? Just because it looked different?
Pretty much. The reasoning was based purely on physical observation. Morphology.
Because of that thick ring we just talked about.
Exactly. Because it had that prominent membranous ring on its stem rather than a flimsy cobweb cortina, early taxonomists believed it was just too structurally different to be a true web cap.
It just didn't fit the visual mold.
Right. However, the advent of modern genetic testing completely flipped that. Specifically, the sequencing of the ITS regions of fungal DNA.
The IT?
Yeah, the internal transcribed spacer regions. It's a sort of genetic barcode for fungi. And it completely revolutionized our understanding of fungal relationships.
So basically, DNA sequencing allowed scientists to redraw the family tree based on actual genetics, rather than just what the mushroom looked like to the naked eye.
Exactly the case. When researchers sequenced the DNA of Rosites caperata, the phylogenetic data was undeniable.
It was a web cap all along.
It nested firmly deep within the evolutionary lineage of the Cortinarius family. The prominent ring was essentially just an evolutionary variation of the Cortina, not a sign of an entirely different genus.
Wow. So nature just built a thicker veil.
Pretty much. Consequently, the scientific community officially changed its name to Cortinarius caperatus, reflecting its true genetic heritage.
There is also a notable cultural shift regarding its name that the sources highlight.
Yeah, yes, the common name.
Right. For generations, traditional foragers referred to it as the gypsy mushroom.
You'll still see that in a lot of older printed field guides.
You definitely will. But modern nomenclature databases have moved away from that term. Around 2020, major mycological platforms like iNaturalist and NatureServe officially transitioned the accepted common name to the wrinkled Cortinarius, or wrinkled cort.
Which makes a lot of sense from both a cultural and scientific standpoint.
Yeah, it mirrors similar efforts in other scientific fields, like the American Ornithological Society renaming certain birds. The goal is to update legacy names that rely on derogatory or culturally insensitive terms, focusing instead on descriptive names that actually help identify the organism.
And wrinkled cort tells you exactly what to look for. The wrinkled cap.
Exactly. So the synthesis of all this information paints a very appealing picture for a wild food enthusiast.
It does. It sounds ideal.
We have a robust, easily identifiable, choice edible mushroom that grows in abundance across vast stretches of the Northern Hemisphere, backed by modern genetic science.
It sounds like the absolute perfect target for a forager. A delicious, widespread woodland treat.
So this is where the story takes a sharp turn into the invisible and the hazardous.
Yes, this is where things get complicated.
Because this specific mushroom, more than almost any other, acts as a hyper-efficient environmental sponge.
To understand the scale of what it absorbs, we really need to dive deep into a massive longitudinal study conducted in Poland between 1996 and 2016.
Right, and alongside that, we have recent monitoring data published by the International Atomic Energy Agency, the IAEA, focusing on the forests of Bavaria.
These studies force us to confront the long-lasting environmental legacy of the 1986 Chernobyl nuclear disaster.
It's crazy that an event from 1986 is still directly impacting our food in the 2020s.
It is, but when reactor number 4 at the Chernobyl nuclear power plant experienced a catastrophic core meltdown and explosion, the intense heat and fire lofted massive quantities of radioactive isotopes high into the atmosphere.
Just a giant cloud of radiation.
Yes, and the prevailing wind patterns carried this radioactive plume across vast swaths of the European continent.
So it wasn't just Ukraine that got hit.
Not at all. When it rained, a process known as wet deposition washed these microscopic radioactive particles out of the clouds and onto the soils of countries hundreds of miles away.
Including Poland and southern Germany.
Exactly. And one of the most abundant and persistent of these isotopes is radiocaesium, specifically cesium-137.
Cesium-137. And that is the focal point of the Polish study published in the journal Environmental Pollution.
Yes. The researchers set out to measure exactly how much of this specific radioactive isotope was present in wild fungi.
And they focused heavily on our mushroom, the wrinkled cort.
They did, and the numbers they reported are staggering.
How did they even go about testing this over such a long period?
The methodology of the Polish study was exhaustive. Over a 20-year period, researchers took physical samples of Cortinarius caperatus fruiting bodies.
So the mushrooms themselves.
Right, alongside core samples of the underlying forest topsoil. And they did this across 22 different geographical locations in northern Poland.
That's a lot of data points.
It is. They brought these samples back to the lab, dehydrated the mushrooms to remove the highly variable water weight, and used gamma-ray spectrometry to measure the radioactive emissions.
Gamma-ray spectrometry. So they're literally counting the radiation coming off the dried mushrooms.
Exactly. And what they documented was that decades after the initial Chernobyl fallout had settled, the wrinkled cort was still pulling immense concentrated amounts of radiocaesium out of the forest floor.
To put the scale of those measurements into perspective, we have to look at the legal safety limits.
Right, we need a baseline.
The European Union heavily regulates the amount of radiocaesium allowed in market food. That strict legal limit is 600 becquerels per kilogram of fresh weight for adult consumption.
Okay, let's break down that unit. A becquerel.
Yeah, a becquerel is the standard scientific unit of radioactivity. It simply means one atomic nucleus is decaying and emitting a particle of radiation every single second.
So 600 decaying nuclei per second per kilogram of food.
Exactly. That is the absolute maximum the EU considers safe to eat.
Keep that number in mind. 600.
So in the Polish study, measuring the dry weight of the mushroom caps, the activity levels spiked up to 18,000 becquerels per kilogram in the year 2002.
18,000.
18,000. That is 30 times the EU limit.
It's an incredible concentration, and the problem is not confined to Poland.
No, the IAEA data from Bavaria, collected much more recently between 2018 and 2020, notes that there are still localized hotspots in the forest soil containing more than 100,000 becquerels of cesium-137 per square meter.
100,000. That's just wild. But the defining scientific question that emerges from this data is the timeline.
Right, because the Chernobyl disaster occurred in 1986.
Yes, yet the Polish researchers observed the radiation levels in these specific mushrooms actually rising throughout the 1990s and peaking in the early 2000s.
That is the most counterintuitive part of the whole data set.
It really is. Why were the mushrooms getting hotter, more radioactive, a decade or more after the initial contamination event?
You would naturally assume that radiation from a single event would just, slowly fade away, like wash out into the rivers or just naturally decay over time.
You'd think so, yeah.
Finding that the mushrooms were actively increasing their radioactive payload years later seems to defy basic physics.
It does seem weird. But the answer lies in the intersection of soil mechanics and fungal architecture. It is a slow-motion ecological process.
Okay, walk us through it. What's happening underground?
Well, when the radioactive fallout first rained down in 1986, it was intercepted by the very top layer of the forest floor.
The O horizon, right.
Exactly. The O horizon, which consists of freshly fallen leaves, pine needles, and raw organic humus.
So just the surface litter.
Yeah. And at that moment in 1986, mushrooms that feed exclusively on surface litter became highly radioactive.
Because the radiation was right there on the top.
Right. But cesium-137 is water-soluble.
Ah, so rain changes everything.
It does. Over the ensuing decades, every time it rained, the water dissolved tiny amounts of that surface cesium and carried it millimeters deeper into the earth.
So it was migrating.
Yes, migrating slowly down into the underlying mineral soil layers, the A and B horizons.
So the radioactive contamination is basically slowly sinking. It's creating this descending layer of toxicity moving through the earth.
Let's visualize the soil column as an ocean. And the radiation is like a dense cloud of debris slowly sinking from the surface down into the depths.
Okay, I can picture that.
Now, the mushroom you see above ground is merely the temporary fruiting body. It's the apple on the tree.
The real organism is underground.
Exactly. The permanent organism is the mycelium, a vast perennial network of microscopic thread-like tubes called hyphae.
And different mushrooms live at different depths, right?
Right. Different species of fungi establish their mycelial networks at different highly specific depths in the soil ocean, depending on where they find the best nutrients.
And where does the wrinkled quart live?
Cortinarius caperatus happens to possess deep penetrating mycelia. It builds its primary network deep down in the mineral soil horizons.
Oh, okay, so tying the physics to the biology, the deep mycelial network of the wrinkled quart is sitting down there like a deep sea net.
Exactly.
For the first few years after Chernobyl, the radiation was mostly above it, trapped in the surface leaves.
Yep.
But as the rain slowly pushed that radioactive layer deeper and deeper over the years, it eventually descended right into the exact feeding zone of the wrinkled quart.
That is the precise mechanism. The radioactive wave took years to slowly percolate down to the specific depth where the wrinkled quart does its feeding.
So it wasn't the mushroom changing its appetite.
No, the mushroom wasn't changing its behavior at all. The environment was just delivering the 1986 fallout to its front door a decade later.
That is mind-blowing. And this study gets incredibly granular about how the mushroom handles this material once it absorbs it.
They did some really fascinating anatomical breakdown.
Right, the researchers didn't just blend the whole mushroom up and test it. They separated the caps from the stipes, the stems, and tested them individually.
And they found a stark difference.
Yeah, the caps hold significantly more radiocaesium than the stems, with an average ratio of about 2.4 to 1.
Which means the reproductive cap is the primary storage zone for the radiation.
But why? To understand the biochemical significance of that ratio, the researchers needed a control variable, right?
They did. They needed to prove that the mushroom was actively manipulating the cesium, not just passively soaking up contaminated groundwater like a paper towel.
So what do they use as a control?
To do this, they measured the concentration of a naturally occurring non-toxic stable isotope, potassium-40.
Potassium, which is something the mushroom actually wants.
Yes. Potassium is a vital, essential macronutrient for all fungi. They require it to regulate internal osmotic pressure.
Meaning they use it to manage their water content and keep their cells inflated.
Exactly. When the researchers measured the potassium-40 levels in the wrinkled quart, they found it was distributed uniformly. The ratio between the cap and the stem was almost exactly 1-to-1.
The physiological implications of that are astounding.
It really shows how sophisticated they are.
Yeah, the mushroom is actively distributing the essential potassium evenly throughout its entire structure to maintain basic life functions.
Right.
But it is taking the radioactive cesium and deliberately funneling it upward, concentrating it specifically in the cap.
The biological machinery required to sort and transport elements like that is incredibly complex.
But wait, why does the mushroom absorb the cesium in the first place if it doesn't even need it? Does it use the radiation for something?
Not at all. It is a classic case of mistaken chemical identity at the cellular level.
Mistaken identity.
Yeah. Fungi have evolved incredibly sophisticated transport proteins and binding ligands in their cell membranes. These are designed to hoover up essential trace elements from the soil.
So they're like little microscopic scavengers.
Exactly. Now, if we look at the periodic table of elements, potassium and cesium are both alkali metals. They're located in group 1.
Oh, I see where this is going.
Yeah, they share similar chemical properties, similar valencies, and comparable ionic radii.
Meaning they look practically identical to the mushroom cells.
Exactly. Because of this structural similarity, the fungal transport proteins just cannot tell the difference between the potassium they desperately need and the radioactive cesium-137 that is contaminating the soil.
Wow. So the mushroom's biological filters are essentially tricked.
Tricked completely. It absorbs the cesium, assuming it is potassium, and sequesters it in the developing fruiting body.
That biological blind spot, this inability to distinguish between essential nutrients and toxic mimics, is actually a perfect transition into the next phase of our deep dive.
Because radiation isn't the only thing taking advantage of those filters.
Exactly. It is not just nuclear fallout that exploits this fungal machinery. The exact same transport proteins that mistakenly pull in cesium also make the wrinkled quart a highly efficient scavenger of heavy metals.
Let's examine another major study provided in our sources, this one focusing on the accumulation of cadmium and lead in wild edible mushrooms.
Right, let's look at cadmium. Where was this study done?
This particular study was conducted in the eastern region of Poland, an area geographically and culturally known as the Green Lungs of Poland.
The green lungs. That sounds pristine.
It is. This region, encompassing the Podlaskie province, is defined by its vast, dense tracts of primeval forests. It has an incredibly low population density.
So not a lot of cities or traffic.
Right. And most importantly for the study, there's a complete historical absence of heavy industry. There are no smelters, no massive chemical plants, no industrial-scale waste runoff.
Okay, so it is generally regarded as one of the most pristine, unpolluted natural environments in Central Europe.
Absolutely. If a modern consumer was looking for the purest, most organic, untouched wild food possible, the Green Lungs region is exactly the kind of environment they would seek out.
It's the dream scenario for an organic forager.
Yeah. The researchers in this study collected and tested 21 different species of mushrooms from these supposedly pristine forests.
And they needed a baseline again, right?
They did. They also tested commercially cultivated species, like the standard white button mushroom you buy at the grocery store, to serve as a baseline comparison.
Okay, so wild mushrooms from a pristine forest versus grocery store button mushrooms. What were the laboratory results for our main character, the wild wrinkled quart?
Well, the study refers to it by its older scientific name, Rosites caperatus, but the results were highly alarming.
How bad?
The data revealed that even in this completely unpolluted, low-industry environment, the wild wrinkled quart accumulated exceptionally high levels of cadmium.
And we get the numbers on that?
The laboratory analysis found an average concentration of 9.29 micrograms of cadmium per gram of dry mushroom mass.
9.29 micrograms. And what about the grocery store mushrooms?
The cultivated white button mushrooms grown in controlled agricultural compost showed near zero heavy metal levels, averaging around 0.09 micrograms of cadmium.
Okay, wait. 9.29 versus 0.09. The wild wrinkled quart, growing in a pristine forest, was hoarding roughly 100 times more cadmium than the mass-produced grocery store variety.
100 times more, yes.
I have to pause here and ask for some clarification on the environmental logic. Let's debate the origin of this toxicity.
Sure.
If the Green Lungs region is famously unpolluted, if there is no industrial waste, no factories spewing emissions, and no polluted rivers, where's the cadmium actually coming from?
It's a great question.
I mean, you cannot squeeze blood from a stone, and a mushroom cannot extract heavy metals from perfectly clean dirt. Are we entirely sure this isn't the result of distant atmospheric pollution blowing in, similar to the Chernobyl fallout?
It is a crucial distinction to make between man-made pollution and geological reality.
Geological reality.
Yeah. While distant atmospheric transport can account for trace amounts of surface deposition, the primary answer lies in the Earth's crust itself. Cadmium is a naturally occurring element.
Oh, I see. It's just part of the dirt.
It is a fundamental constituent of the planet's geology. It exists everywhere in microscopic, diffuse trace amounts within the soil matrix, entirely independent of human activity.
Okay, so the soil in the green lungs is not polluted in the human sense. It simply contains the baseline geological presence of cadmium.
Exactly. The issue isn't that the forest floor is a toxic waste dump.
The issue is that the mushroom is an aggressive overachiever at finding and collecting what is already there.
That is the exact mechanism at play. Fungi act as powerful biological magnets.
How do they manage to collect so much of it if it's so diffuse in the soil?
Certain species, notably the wrinkled court and several members of the Boletus family, possess specialized genetic adaptations for surviving in nutrient-poor soils.
Okay, what kind of adaptations?
They synthesize specific metal-binding proteins, things like metallothioneins or phytochelatins.
And what do those proteins do?
Well, as their vast mycelial networks spread out over hundreds of square meters underground, they relentlessly scavenge the soil. And they find these microscopic, naturally occurring trace amounts of cadmium.
And the proteins grab it.
Yes, the proteins bind to the cadmium ions, pulling them out of the soil chemistry and concentrating them into the dense tissue of the fruiting body.
So the mushroom is essentially executing a microscopic mining operation.
That's a perfect way to describe it. It's mining the soil. Therefore, even in a forest entirely untouched by human industry, the mushroom manages to present a highly concentrated, potentially dangerous dose of the heavy metals native to the local geology.
This actually brings up a really interesting regulatory conflict.
Oh, the legal side of this is fascinating.
Yeah. Earlier, we discussed how the European Union heavily regulates the radiocaesium levels in wild mushrooms. They set that strict cap at 600 becquerels per kilogram for commercial sales.
Right.
The implication is that radiation is an acute, heavily monitored threat. But here we have cadmium, a highly toxic heavy metal, occurring at alarming levels in the exact same mushrooms.
Sometimes even more alarming levels, functionally.
So why doesn't the discovery of 100 times the baseline cadmium trigger the same level of regulatory panic or market bans as the Chernobyl radiation? Are heavy metals somehow considered less dangerous if they are natural?
The disparity in regulation really comes down to the difference between acute public perception and the chronic nature of heavy metal toxicity. Plus, you have to factor in the complexities of agricultural law.
Okay, explain that.
Well, radiation from a nuclear disaster is a highly visible, politically charged event. It scares people immediately.
It's an invisible killer.
Exactly. So regulatory bodies establish strict, legally binding limits for radiocaesium across all food sectors almost immediately after Chernobyl. They had to manage acute public health fears and monitor the fallout plume.
But heavy metals don't get that same treatment.
No, because heavy metals are pervasive. Because cadmium is naturally occurring in the crust, it is present in almost all soil-grown agriculture to varying degrees. Wheat, potatoes, root vegetables, they all have some trace amount.
So regulating it out of existence is impossible without banning farming altogether.
Exactly. You cannot mandate a zero-tolerance policy for an element that is fundamental to the Earth's crust.
So how do they handle it then?
Instead, health organizations utilize guidelines like the Provisional Tolerable Monthly Intake, or PTMI.
Provisional tolerable monthly intake. So it's an advisory metric rather than a strict market ban.
Right. Cadmium toxicity is insidious because it is cumulative. It does not kill you overnight.
So you don't eat one mushroom and drop dead.
No. Instead, the human body struggles to excrete it. It takes a very long time to leave your system, so it accumulates in the liver and kidneys over decades.
And what happens when it accumulates too much?
It eventually leads to severe renal dysfunction and the demineralization of the skeletal system.
Demineralization. Like your bones get weak.
Yes. It's a condition famously documented in Japan as Itai disease. It's incredibly painful.
Wow.
So the regulatory approach relies more on dietary advice than strict market confiscation. They advise populations to vary their diets to avoid chronic bioaccumulation.
And the Polish study directly addresses that dietary advice for the listener who might be foraging.
They ran the math on it, yeah.
Right. The researchers calculated the potential dietary intake based on their findings. They concluded that eating a single large hearty meal of these specific hyperaccumulating mushrooms like the wrinkled court could easily provide a dose of cadmium that exceeds your entire provisional tolerable monthly intake in one sitting.
One meal, and you've hit your limit for the month.
That is wild. It really reinforces the reality that natural and wild do not automatically equate to safe or healthy.
Not at all. The wild environment is a complex chemical landscape,
So we are left with a culinary puzzle.
a very tricky one.
We have this delicious, widespread mushroom that is secretly packed with Chernobyl radiocaesium and geological cadmium. The immediate practical question for any chef, home cook, or forager is, can I just cook the danger away?
It's the first thing everyone asks.
If I boil it, fry it, or stew it, does the heat neutralize the threat? Let's unpack the culinary chemistry detailed in our sources, because it reveals a fascinating and frankly slightly terrifying paradox regarding how we prepare wild food.
The answer requires us to look at the thermodynamics of cooking. We have to understand how different preparation methods physically interact with both the cellular structure of the fungal tissue and the elemental nature of the toxins locked inside.
Right. So can heat just destroy the cadmium or the cesium?
No, we cannot destroy heavy metals or radioactive isotopes with the heat of a kitchen stove.
Because they're basic elements.
Exactly. They are base elements, not complex bacterial proteins that denature when cooked. You can't cook away metal. However, we can manipulate where they end up.
Okay, let's begin with the process of blanching.
The Polish radiation studies specifically investigated how different preparation methods affect the final radiocaesium levels before consumption. And they found that a rigorous blanching process is highly effective.
How rigorous are we talking?
If you parboil the fresh mushrooms in a large excess of vigorously boiling slightly salted water for 10 to 15 minutes.
Okay, so boiling them for a good while.
Yes. And then crucially, throw that boiling water down the drain, you can successfully remove up to 90% of the radiocaesium from the mushroom tissue.
90%, that's huge. What is actually happening there physically?
The physical process occurring there is leaching.
Like making tea.
Exactly like making tea. The sustained heat of the boiling water breaks down the chitinous cell walls of the mushroom. And because cesium is highly water-soluble, remember how it migrated through the soil with the rain? The internal fluids of the mushroom mix with the boiling water.
And the cesium just moves into the water.
Yes. The cesium ions freely leach out of the tissue and into the surrounding liquid phase. By discarding that water, you are physically washing the radioactive isotope out of your meal and down the sink.
Okay, that sounds like a great solution. But, and this is the massive consequential but, in the sources, if you prepare the mushrooms the way the vast majority of culinary enthusiasts actually want to eat wild fungi, you run into trouble.
Oh, big trouble.
Which is pan-frying them in butter, sautéing them in oil, or slowly stewing them in their own juices. If you do that, you achieve the exact opposite result.
You do.
Frying or stewing actually increases the relative concentration of the radiation and the heavy metals in the final dish.
It seems totally counterintuitive, but from a physical and thermodynamic standpoint, it makes perfect sense.
Explain the physics of frying them then.
Well, pan frying applies high heat, and that high heat causes the extensive water content within the mushroom to rapidly release and evaporate into steam.
And mushrooms are mostly water.
Yeah.
Exactly.
They can be up to 90% of its mass. So as that water steams off, the physical tissue of the mushroom shrinks, dehydrates, and condenses.
But the metals and the radiation don't evaporate.
No, they do not evaporate at cooking temperatures. They remain entirely locked within the shrinking cellular matrix of the mushroom, or they end up dissolved in the cooking fats left in the pan.
It is the exact same principle as making a culinary reduction sauce.
That's a great analogy.
When you simmer a stock, the water evaporates, leaving behind a much smaller volume of liquid with a massively intensified, concentrated flavor profile.
Right.
Except in this scenario, instead of concentrating the umami flavor of chicken stock, you are actively concentrating the geological cadmium and the Chernobyl radiation.
You really are.
You are taking a baseline level of toxicity spread throughout a large water-filled mushroom and packing it into a smaller, denser, much more potent bite of food.
That is a highly accurate representation of the chemistry. You are increasing the toxic payload per gram of food consumed.
And this culinary reality connects directly back to the regulatory landscape we debated earlier.
Yes, the EU limits.
Right. We noted that the European Union maintains a strict legal limit of 600 becquerels per kilogram for market food.
So if a commercial foraging operation harvests these mushrooms from the Bavarian forests and attempts to sell them to a restaurant or a grocery store, that batch is legally subject to radiological testing.
And if they test too high?
If the fresh weight exceeds the 600 becquerel limit, the entire harvest is confiscated and banned from the commercial market to protect the public.
But the laws do not extend into the private kitchen.
Not at all. Private recreational foragers operate in a completely unregulated space regarding consumption. The government will not protect you from your own harvest.
If you go out into the Bavarian woods or the pristine forests of the Polish green lungs, pick a basket of wild wrinkled quartz, take them home and fry them in butter, there is no health inspector standing in your kitchen checking your radiocaesium levels.
You are on your own. It is entirely incumbent upon you, the individual forager, to understand the deep geological history of your local forest and the chemical consequences of your cooking methods.
Does the IAEA offer any advice for people living there?
They do. The IAEA explicitly states in their literature that individuals living in contaminated regions should proactively inform themselves. And if they wish to keep their internal radiation exposure minimized, they should strictly avoid the regular consumption of these known hyperaccumulating wild species.
So basically just don't eat them often.
Or at all, if you can avoid it. Which brings us to a critical transition in our understanding of wild foraging.
The legal risks.
Yes. While federal and state governments may not regulate what you choose to eat out of your own personal foraging basket, they absolutely, strictly, and aggressively regulate where and how you are allowed to collect that basket in the first place.
The biochemical risks of heavy metals and radiation are only half of the story. The legal risks of physically picking the mushroom are equally complex and potentially much more immediate.
Oh, absolutely.
This is where the modern legal minefield of foraging really opens up, because according to our foraging guide source, recreational mushroom foraging is no longer a niche, obscure hobby.
It has exploded in popularity.
It has experienced an explosive boom, growing by over 300% in just the last decade in the United States alone.
300% is massive.
It is. Millions of new enthusiasts are heading into the woods armed with field guides and baskets. And millions of people are walking face first into a dizzying, overlapping patchwork of federal, state, and local land management laws that are incredibly difficult to navigate.
The foundational issue that causes so much confusion is jurisdiction.
Jurisdiction, right. Who owns the land?
Exactly. The United States contains hundreds of millions of acres of public land. But that land is not a monolith. It is divided and managed by several different federal agencies, each operating under entirely different legislative mandates and philosophical approaches to conservation.
So the rules change depending on whose land you're standing on.
The rules regarding what you can and cannot take from the forest change the millisecond you cross an invisible boundary line on a map.
Let's break down those federal agencies, starting with the most restrictive, the National Park Service, the NPS.
They manage roughly 85 million acres of iconic American landscapes.
And they operate under a very specific mandate.
Yes, the Organic Act of 1916, which is focused on strict, unyielding preservation.
Preservation over use.
Exactly. The goal is to keep the ecosystem exactly as it is, unimpaired for future generations. Consequently, foraging of almost any kind is strictly prohibited in nearly all national parks and national monuments.
So you can't just pick a mushroom off the trail in Yellowstone.
No. You cannot pick mushrooms, you cannot harvest wild onions, you cannot take interesting rocks home.
Now, the sources do note some rare, highly specific exceptions, right?
Yes, primarily for subsistence gathering by local indigenous residents in certain massive Alaskan parks. But the general rule of thumb is absolute. If you are standing in a national park, keep your hands in your pockets.
Conversely, you have agencies like the USDA Forest Service, which manages 193 million acres of national forest.
And the Bureau of Land Management, the BLM, which oversees 245 million acres of public lands, mostly in the West.
And they have a different philosophy entirely.
They do. These agencies operate under a fundamentally different philosophy dictated by the Multiple Use Sustained Yield Act of 1960.
Multiple use, meaning recreation, logging, that kind of stuff.
Right. Their mandate requires them to balance conservation with public recreation, timber harvesting, grazing, and resource extraction.
And because their legislative DNA includes resource extraction, national forests and BLM lands are generally the most accessible and friendly environments for recreational foragers.
They are, however, friendly is not a synonym for a free-for-all.
Right. You can't just back up a truck and clear out a forest.
Exactly. Even within the more permissive national forests, there are strict quotas and limits.
What's the typical limit for a casual forager?
For casual personal use, the limit is typically capped at 1 to 5 gallons of mushrooms per person per day, though this varies wildly depending on the specific ranger district.
And in many highly trafficked areas, particularly in the Pacific Northwest where mushroom hunting is culturally ingrained, You are still required to stop at the ranger station and obtain a free personal use permit before you even step foot into the trees.
Which is an extra hurdle, but it's important. What is absolutely crucial for the modern forager to understand, though, is the rigid legal distinction between personal use and commercial use.
Because the judicial system treats these two activities with vastly different levels of severity.
Vastly different.
Yeah.
Crossing the line from personal to commercial is easier than most people realize.
So personal use is legally defined quite narrowly. You are taking the harvest home to consume it yourself, or to share it with your immediate family and friends.
Right. No currency, goods, or services change hands.
Commercial use, on the other hand, is defined as any form of sale or barter.
Any form, even small scale.
And here is the modern trap that catches a lot of enthusiastic amateurs. The regulatory sources explicitly warn that selling even a handful of excess mushrooms to a coworker or trading a basket of chanterelles for a haircut instantly reclassifies you.
Oh, absolutely. Or, most commonly, posting a photo of your massive haul on social media and offering to sell the surplus locally.
The federal government considers that a commercial operation.
Yes, and it is vital to note that law enforcement agencies within the Forest Service actively monitor local social media marketplaces and foraging groups.
Wait, they monitor Facebook groups?
They do, specifically to identify unlicensed commercial activity. If you are operating commercially, the regulatory requirements skyrocket immediately.
What do you have to do?
You are legally required to purchase commercial harvesting permits, which can range from $50 to over $400 per season, depending on the target species.
That's a steep jump.
And you may be restricted to highly specific, rotating harvest zones to prevent localized depletion. Furthermore, commercial foragers are often mandated to register the specific buyers they sell to and submit detailed end-of-season harvest reports.
So you have to literally document exactly how much biomass you remove from the forest.
Exactly.
And if an individual decides to ignore these complex rules, the penalties outlined in the legal sources are severe.
Very severe. This is not a scenario where you get a polite warning and a slap on the wrist.
If you are caught foraging in a strictly closed area, such as a national park or a designated scientific research zone within a national forest, the fines can reach up to $5,000.
$5,000.
And federal magistrates have the authority to impose up to 6 months in jail.
Just for picking a mushroom in the wrong place.
And it gets worse. If you are caught engaging in commercial poaching, which means harvesting large quantities with the intent to sell without the requisite commercial permits, the financial penalties can hit a staggering $20,000.
$20,000.
Furthermore, law enforcement officers possess the authority to immediately confiscate your vehicle, your harvesting equipment, and your entire haul as evidence of the crime.
They can take your car.
Right.
To understand the severity of these penalties, we really must look at the ecological reasoning behind the laws.
Right. These regulations are not born of bureaucratic overreach or a desire to ruin a weekend hobby.
No, they are fundamentally necessary tools for ecosystem protection and the sustainable management of our natural resources.
This circles back perfectly to the biology we discussed at the beginning of the episode. It is all about protecting the role the mushroom plays in the broader forest ecosystem.
Precisely. Recall the mycorrhizal networks we detailed. That vital symbiotic relationship where the fungal mycelium provides essential water and minerals to the roots of the forest trees.
Right, the underground root network.
The mushroom that a forager plucks from the ground is the reproductive organ of that vast, hidden underground network. Its sole biological purpose is to mature, open its cap, and drop millions of spores to propagate the next generation of fungi across the forest.
And if you take it away before it drops those spores.
Exactly. If a specific area is aggressively and repeatedly overharvested, particularly by unpermitted commercial crews sweeping through a forest in a grid pattern to take every single valuable mushroom, they see it physically removes the reproductive bodies from the ecosystem before they ever have a chance to complete their life cycle.
And without the dispersal of spores, there is no genetic diversity and no next generation of fungi to colonize new tree roots.
And if the fungal population crashes, the mycorrhizal network degrades.
So the trees suffer.
The surrounding trees lose their symbiotic life support system, which severely compromises the health, growth, and drought resistance of the entire forest canopy.
Not to mention the animals.
Right. Wild mushrooms serve as a critical nutrient-dense food source for a wide variety of wildlife, from deer and wild boar down to insects and rodents.
So by enforcing strict daily limits and requiring permits, The land management agencies are actively attempting to ensure that enough fungal biomass remains in the woods to sustain both the wildlife and the invisible subterranean architecture of the forest itself.
It's all connected.
And when you dig into the state-by-state data provided in the legal guides, the jurisdictional maze becomes even more granular and intimidating.
The state laws are a whole other layer of complexity.
In the western United States, a state like California explicitly prohibits foraging in its state parks, viewing them as preservation zones, but they actively allow and manage foraging in its state forests.
Right, so even within one state, the agency matters.
In Washington and Oregon, which are the epicenters of a massive multi-million-dollar commercial wild mushroom trade, especially for highly prized, export-driven species like the matsutake, the regulations are incredibly tight.
They have to be to manage the commercial pressure.
They utilize tightly controlled special harvest zones, strictly enforced daily commercial quotas, and mandatory buyer reporting to track the biomass leaving the woods.
Very heavily regulated.
Meanwhile, in the Midwest, a state like Michigan is culturally noted as being highly foraging-friendly on its state lands, particularly during the explosive, deeply traditional spring morel hunting season.
Oh, morel season is huge there.
But if you drive an hour south and cross the border into Ohio or Indiana, foraging on state-owned public land is largely prohibited by default. You have to know exactly where you are standing on the map at all times.
This overwhelming variability is exactly why the primary universal advice across all regulatory guides is proactive communication.
Just calls her.
Call them. Before you step into the woods with a basket, you must contact the specific local land management agency, the exact ranger district office, or the local BLM field manager responsible for the exact parcel of land you intend to walk on.
Because the rules don't just vary by state and federal agency. They can change dynamically from year to year.
Or even month to month. A ranger might close a previously open forest road due to extreme fire danger. They might restrict access to a specific valley to protect sensitive wildlife migrations. Or temporarily ban harvesting in a certain area due to localized concerns over recent overharvesting.
You really just have to ask. Let's take a deep breath and recap the rather intimidating situation surrounding our main character, the wrinkled court.
It's a lot to process.
We have established that it is a significant legal hassle just to figure out where you can legally walk in the woods to pick it without risking thousands of dollars in fines.
Check.
If you successfully navigate the legal maze and harvest it, the mushroom might be heavily laced with naturally occurring geological cadmium because its mycelium acts like a biological mining operation.
Check.
And if you happen to be foraging in Central Europe, the mushroom might literally be ticking with radiocaesium from a decades-old nuclear meltdown. A toxic payload that you can accidentally condense and concentrate if you choose to fry it in butter rather than blanch it.
When synthesized, the data paints a highly complex, scientifically fascinating, but undeniably intimidating picture for the casual culinary forager.
It really does. It makes you question whether the butter-sautéed meal is worth the background anxiety.
I wouldn't blame anyone for opting out of eating it.
So, what is the alternative? If you are fascinated by the massive Cortinarius family, but you are terrified to eat them because of the latent kidney toxins and the deadly cousins, the heavy metal bioaccumulation, or the lingering ghosts of Chernobyl, is there any other way to appreciate these organisms?
Thankfully, yes.
What can you actually do with them?
This brings us to a wonderfully creative, entirely safe, and historically rich application of the Cortinarius genus. If you decide that the toxicological risks of ingestion are too high, you do not have to leave them in the woods. You can use their incredibly complex internal chemistry to paint your world. They are massively popular, highly prized resources among the global community of natural dye enthusiasts.
Dyeing, like clothing. I find this application absolutely brilliant.
It's fantastic.
Our sources detail how Cortinarius mushrooms, despite often looking like drab, brown, uninteresting lumps on the forest floor, possess the chemical potential to produce a mind-blowing spectrum of vibrant, permanent colors for textiles.
The colors are unbelievable.
And the physical process of extracting that color is relatively straightforward, right?
It is. The literature describes simmering the freshly harvested or dried mushrooms in a pot of water to draw out the water-soluble pigments.
Just boiling them down.
Yeah, but to ensure the dye permanently binds to the fibers of the yarn, wool, or silk, dyers utilize a chemical compound known as a mordant.
A mordant? What exactly does that do?
The chemistry of mordanting is essential to the process. A mordant, typically a metallic salt like alum, which is potassium, aluminum sulfate, or iron or copper, acts as a chemical bridge.
A bridge between the mushroom and the fabric.
Exactly. It forms a strong coordination complex between the dye molecules extracted from the mushroom and the protein molecules in the animal fibers. Without the mordant, the color would simply wash out the next time the fabric got wet.
But with the mordant, the dye becomes color fast.
Yes, and the specific biochemical compounds hidden within the Cortinarius species, specifically a class of organic pigments known as anthraquinones, yield a remarkably brilliant color palette.
The color spectrum documented in the dyeing guides is stunning. While the external skin of many webcaps is a muted tawny brown, the internal chemistry tells a different story.
It really does.
For example, there is a species appropriately named Cortinarius sanguineus, the blood-red webcap. When simmered, it produces intense, deep crimson and brilliant blood-red dyes that rival anything produced synthetically.
The diversity of color across the genus is a testament to their chemical complexity. If you harvest Cortinarius archery, a prominent species found in Australia, the extraction process yields vibrant, deep greens and rich olive tones.
Green from a mushroom, that's so cool.
And Cortinarius purpurescens contains pigments that can dye protein fibers, beautiful shades of purple and deep blue violet. And Cortinarius scarcius provides brilliant, sunlit golden yellows.
So you can get a whole rainbow.
You can. Furthermore, the exact final shade a dyer achieves is highly variable. It can shift dramatically depending on the maturity of the mushroom when it was picked, the specific metallic mordant used.
So different mordants change the color.
Yeah. Iron might darken a red into a deep burgundy, while alum keeps it bright. And it also changes based on whether the dye is applied to coarse sheep's wool or fine silk. It is as much an art form as it is organic chemistry.
Utilizing these mushrooms for natural dyes is a perfect example of leveraging the biochemical richness of fungi in a way that entirely bypasses the toxicological risks of eating them.
It's the perfect workaround.
You are taking the exact same complex internal chemistry that makes them such efficient, dangerous absorbers of trace heavy metals and using it to extract stable, vibrant, beautiful pigments.
It allows individuals to engage deeply with the ecology of the forest, to forage, and to utilize wild resources without ever having to worry about the provisional tolerable monthly intake of cadmium or the latent kidney damage of orellanine.
It is a completely safe, highly rewarding interaction with a notoriously difficult genus of fungi.
Absolutely.
If you are out in the woods navigating the legal boundaries of a national forest and you stumble upon a massive, beautiful patch of webcaps, you don't have to walk away in fear.
No, you can appreciate them.
Even if the heavy metal data and the radiation charts we unpacked today have thoroughly convinced you never to put a wild Cortinarius in your frying pan, you can still legally and safely harvest a few to dye a scarf or a wool sweater, a brilliant, natural blood red or vibrant green.
It is the perfect creative alternative to culinary risk.
To bring this expansive journey all together, we started our deep dive focused on Cortinarius caperatus, the wrinkled court, evaluating it as a widespread, choice, edible woodland treat.
And it led us down quite a rabbit hole.
It did. We uncovered the hidden biology of its deep penetrating mycelial network and its specialized transport proteins, which combined to turn this simple mushroom into a highly efficient deep earth sponge.
We learned how it indiscriminately hoards geological heavy metals like cadmium from pristine soils.
And acts as a long-term biological trap for nuclear fallout, pulling radiocaesium from historical disasters like Chernobyl up to the surface decades after the event.
We also dissected the thermodynamics of culinary chemistry, revealing the critical difference between leaching toxins out through blanching versus inadvertently concentrating the chemical danger into a potent reduction through pan frying.
We navigated the incredibly complex, highly localized, and heavily enforced legal landscape that governs our public lands. Laws designed to protect the vital, invisible mycorrhizal networks from the devastating ecological impacts of commercial overharvesting.
And finally, we explored how the intricate internal chemistry of the broader Cortinarius family can be safely harnessed to paint textiles in vibrant, natural hues.
It is a stunning amount of scientific complexity, historical legacy, and legal jeopardy hidden inside a single, wrinkled, golden brown mushroom.
When we synthesize all these disparate fields, the botany, the nuclear physics, the legal codes, and the organic chemistry, it leaves us with a profound, overarching realization about the fundamental role fungi play in our global environment.
They really are foundational.
They are not merely passive organisms growing in the dirt. They are highly active, deeply integrated chemical engineers of the forest floor.
I completely agree with that assessment. I want to leave you with a new paradigm to ponder, building on that idea of fungi as active engineers.
It's a great way to reframe how we see the forest.
We are historically conditioned to view trees as the primary record keepers of the forest. We study them, we core them, and we use the physical rings embedded in their trunks to tell us the history of the landscape, documenting ancient droughts, hard winters, or historical forest fires that swept through centuries ago.
Trees are the classic historians.
But perhaps fungi, with their vast subterranean networks and insatiable chemical appetite, are the true, living memory banks of the Earth. When a mushroom like the wrinkled quart absorbs radioactive isotopes from a catastrophic human disaster decades past, or tirelessly pulls ancient, trace heavy metals from deep beneath the crust of the Earth, it is doing more than just surviving.
It's doing something much more profound.
It is literally pulling the forgotten, invisible history of our planet up from the dark, concentrating it, and presenting it to us, visible and tangible right above the ground.
They are the quiet, relentless archivers of the planet's chemistry, forcing us to confront the long-term consequences of both our industrial actions and the natural makeup of our world.
The next time you walk through the woods, take a moment to look down. When you see a mushroom pushing its way up through the damp moss and autumn leaves, ask yourself, what invisible, ancient, or radioactive history is pulsing through the vast hidden network right beneath your boots? Thank you so much for joining us on this deep dive into the source material. Until next time, keep learning and stay curious.